faithfully conveying value and interpreting great books from around the world. Hi, this is Sally with the Book Insights. Today we're diving into the book titled The Drug Hunters. The subtitle is The People Who Discover New Drugs. Since prehistoric times, our ancestors tried chewing roots or leaves to cure illnesses. They were the earliest drug hunters, relying on luck and chance to find the first medicines in nature. Even as science advances, the work of drug hunters, even in modern times, remains full of uncertainties. Salvarsan, used to treat syphilis, was a byproduct of malaria drug research. Vitamin C's discovery came from two guinea pigs accidentally getting sick, and the anti-rejection drug sirolimus was only approved 25 years after its discovery. Today's book, The Drug Hunters, will not only detail the bumpy journey of inventing new drugs, but also fully review the history of drug development. The book's author, Donald Kirsch, is an American biologist with 35 years of drug development experience and 24 drug-related patents. He has served as a research team leader, manager, and chief scientist at several top pharmaceutical companies. The other author, Ogi Ogus, is an American professional science writer who has published many popular science articles in outlets like the Wall Street Journal and the Boston Globe. In this book, they share stories of world-changing drugs like anesthetics and antibiotics, and discuss drug development experiences and challenges based on their work. Next, I'll break down the book's main content into three parts for you. First, what are the origins and development of the drug hunting path? Second, what are the success factors for drug hunters in developing new drugs? Third, what are the challenges and future of drug development? First, let's understand the origins and development of the drug hunting path. The development of drug hunting has gone through three stages: the plant era, the industrialization era, and the modern scientific era. In prehistoric times, everyone was a drug hunter. Our ancestors would chew on roots or leaves they found by chance, hoping these plants would ease their ailments. By the Neolithic era, people were hunting for medicines in jungles, marking the start of the plant era in drug hunting. But in this period, the search for drugs relied purely on luck, and understanding was mixed with superstition and myths. They believed that drinking a flower brew could make them invisible to enemies, inhaling resin powder could read thoughts, and consuming foul root mixtures could cure diseases. They believed that deities created plants and animals to nourish humans, so they saw every part of plants as God's pharmacy, including flowers, roots, seeds, bark, moss, and seaweed, turning them into remedies through harvesting, peeling, grinding, and boiling. Opium is one of the earliest and oldest drugs used by humans. It's the potent extract from poppies, made by crushing unripe poppy fruits, drying the juice, and grinding it into pure opium powder. As early as 3400 BC, the Sumerians started using opium, which was then called the forget-worry drug. By the 2nd century BC, the famous Greek physician Galen recorded diseases opium could treat, like headaches, dizziness, strokes, coughs, abdominal pain, and insect bites. And
When Zhang Qian was sent on a mission to the western regions, opium was introduced to China.
during the Three Kingdoms period, the renowned physician Hua Tuo used cannabis and opium as anesthetics. By the 16th century, pharmacology grew significantly as a branch of botany. German pharmacist Valerius Cortis compiled a pharmacopoeia, meticulously documenting properties and extraction methods for 225 medicinal plants, including ink medicine, saffron, cinnamon, pepper, camphor, and more. However, even though pharmacists mastered plant extraction methods, they didn't know which components were actually effective. In their view, the specific ingredients of plants were unfathomable and impossible to identify. Some scientists even thought there was a mysterious vital force that gave each plant its own soul. By the 19th century, the drug hunting path entered the industrial era. On one hand, chemistry's development explained drug compositions, spurring the birth of synthetic drugs. On the other hand, drug production became scaled and standardized. In the early 19th century, British scientist John Dalton proposed the atomic theory, stating atoms are indivisible chemical particles. Atoms combine through strict mathematical laws to form molecules. With this theory, pharmacists could finally analyze ancient formulas, determine exact molecular structures, and identify the most effective ingredients. For instance, willow extract was used for thousands of years to reduce fever and inflammation, but no one knew which component was key to lowering fever. It wasn't until 1838 that an Italian chemist finally extracted the key active ingredient from willow trees, salicylate. By the 1830s, synthetic chemistry emerged, enabling combination of single chemical elements into complex compounds. The dye industry first utilized synthetic chemistry technology. Just minor formula tweaks produced entirely different colors. Inspired by this approach, Germany's Bayer began modifying known drugs' chemical compositions to create more effective medicines. They first chose salicylate, which reduced fever, pain, and inflammation, but caused obvious side effects like tinnitus and nausea. After experimentation, Bayer synthesized aspirin, similar effects to salicylate, but with significantly reduced side effects. Aspirin became an overnight sensation, remaining one of the most common medicines from the 19th century to today. With mid-19th century industrialization, pharmaceutical production entered standardized mass production. Previously, medicines were sold exclusively through apothecaries, small retail shops. Pharmacists prepared drugs based on their own interpretation and experience, resulting in inconsistent products across different shops. For high-demand drugs like ether, pharmacies couldn't meet demand, and quality varied significantly.
Ether, essential for major surgeries, was extremely difficult to synthesize. Uncontrolled purity made proper dosing impossible for doctors.
In 1854, U.S. Navy doctor Edward Squibb improved ether production by replacing open flames with steam coil heating, achieving standardized manufacturing. Years later, Squibb founded his own pharmaceutical company, renowned for standardized, reliable products in America. The birth of large pharmaceutical companies like Squibb's propelled drug standardization and mass production. After entering the 20th century, drug hunting entered the modern scientific era. Its medical understanding of pharmacology deepened significantly. The emergence of genetic recombination technology brought new inspiration to drug development. Simultaneously, governments strengthened drug safety regulations. Between 1895 and 1930, the medical community had diverse opinions on how drugs worked. One theory suggested drugs worked by altering cells' surface tension. Another theory claimed drug effects depended on dosage. Small doses stimulate, medium doses inhibit, large doses destroy. But in reality, these theories were all incorrect. In 1897, German medical scientist Paul Ehrlich proposed a revolutionary receptor theory. He believed receptors were molecular regions on human cells where drugs interact with specific components to produce therapeutic effects. For example, the asthma drug albuterol reacts with beta-2 receptors, relaxing lung-smooth muscle cells to open airways and reduce asthma symptoms. But this theory faced opposition from scientists and wasn't proven correct until 60 years later. Technological advances not only deepened pharmacological understanding but created innovation opportunities in drug development. The emergence of genetic recombination technology in the late 1970s was such an opportunity. In 1972, Stanford professor Paul Berg inserted bacterial DNA into monkey cell DNA, creating the first recombinant DNA molecule. The bacterial genes altered monkey cells' functioning, enabling them to produce new molecules. For drug developers, reverse genetic recombination was more significant, inserting mammalian DNA into bacteria. In 1975, scientists inserted a hemoglobin-producing rabbit gene into E. coli in a petri dish. The bacterial cells then produced hemoglobin, marking the birth of genetic engineering drugs. Over 10 genetic drugs represented by insulin emerged to treat protein deficiency diseases. With new drugs flooding the market, regulation became crucial. In early 20th century America, drug regulation was weak. Market entry required no safety reviews. Though Congress established the FDA in 1906, it had no real power. The AMA didn't participate in approvals, and laws didn't mandate pre-market animal testing. Under such regulatory loopholes, a severe drug crisis erupted in America in 1937. The Massengill Company developed a new sulfa antibiotic by dissolving sulfanilamide in diethylene glycol, adding raspberry flavoring to create an oral liquid for treating streptococcal infections. To accelerate market launch, they skipped animal testing, failing to discover diethylene glycol's high toxicity, ultimately causing over 100 deaths in America. After the crisis, amid public outcry, Congress passed the 1938 Federal Food, Drug, and Cosmetic Act, requiring FDA oversight of every drug development stage. Pharmaceutical companies must first conduct test tube and animal trials. Only after safety standard compliance can human trials be approved. Drugs require FDA-confirmed safety and efficacy before market approval. Okay, that covers the first part. Drug hunting began in the plant era, developed through industrialization, and evolved into the modern scientific era. Through drug hunters and pharmaceutical companies' efforts, numerous new medicines were developed. Next, in part 2, we'll explore success factors in new drug development. 4 key factors. First, unconventional creativity is fundamental to drug discovery. Salvarsan, an early 20th century syphilis treatment, emerged from a revolutionary concept, attaching toxins to dyes for precision strikes on pathogens like nuclear missiles. In the 1860s, French biologist Louis Pasteur proposed microbes as disease causes. Since bacterial or fungal cells are translucent, staining was needed for observation.
German scientist Paul Ehrlich excelled at cell staining, noticing specific dyes targeted particular cell components while ignoring others.
Each dye seemed biologically target-specific. He then imagined, if a dye targeted pathogens and carried toxicity, it could kill invaders without harming hosts. In 1891, Ehrlich began researching dyes targeting malaria parasites. After testing dozens of dyes, he discovered methylene blue could stain parasites. Even better, this dye itself was toxic to malaria pathogens. Although trials showed it was less effective than quinine for malaria treatment, it proved Ehrlich's theory viable. Ehrlich and his team then tested hundreds of synthetic dyes against parasites, but they struggled to find dyes that could both stain and carry toxicity. Ehrlich then pivoted, deciding to attach known toxins to dye molecules instead. He used arsenic as the toxin for new research and testing. This time, he targeted Trypanosoma cruzi, a parasite causing neurological diseases. He synthesized hundreds of arsenic-loaded dyes and tested them on mice infected with Trypanosoma cruzi. But these compounds either failed to kill parasites or killed the mice too. Feeling defeated, Ehrlich switched to study another disease. He noticed similarities between syphilis pathogens and trypanosomes, actually a misconception. Syphilis is bacterial while trypanosomes are parasitic, differing completely in structure and genetics. Driven by this error, Ehrlich began syphilis research in 1905. The team synthesized 900-plus arsenic dyes, testing on infected rabbits, all failed. In 1907, as they prepared to change strategy, Compound 606, the sixth in the sixth test group, killed bacteria without harming the host. In 1910, named arsphenamine and trademarked Salvarsan, it launched as life-saving arsenic after successful clinical trials. Drug production also required disruptive innovations. Progesterone production exemplified this. Progesterone is a steroid hormone from corpus luteum that inhibits ovulation. Its 1920s discovery sparked scientific interest in fertility research, but extraction was extremely difficult. Animal-derived progesterone was prohibitively expensive. 1 milligram required ovaries from 2,500 sows, and chemical synthesis proved equally challenging. Progesterone is a large steroid molecule that's quite difficult to synthesize. It requires piecing small molecules together one by one, and if you make a mistake, You have to start over. So, how big is this challenge? It's like giving you a pair of scissors and a stack of A4 paper and asking you to piece together a life-sized Great Wall of China. This problem plagued scientists for 20 years. Until 1936, Russell Marker, a brilliant researcher at the University of Pennsylvania in the US, innovatively solved this problem. Instead of synthesizing through traditional addition, he switched to subtraction, meaning degradation, choosing a larger molecule than steroids. Then, removing the unnecessary parts is like finding a piece of paper larger than the Great Wall and trimming it down slightly. Marker first extracted a large molecule similar to steroids from Japanese wild yam tubers and obtained progesterone through degradation. But this yam was small, so progesterone yield was low. He then searched fields for alternatives among over 400 species, but found nothing. Later, he traveled to the hot regions of the American Southwest, heading south, and finally discovered a yam in Mexico with very thick roots. He picked a Mexican yam weighing over 20 kilograms, bribed U.S. customs officials to successfully bring this prohibited plant into the U.S. for experiments, proving it could produce sufficient progesterone. Marker hoped to partner with pharmaceutical companies for mass production, but was rejected. At that time, Mexico was in constant turmoil with high anti-American and anti-wealthy sentiments. All pharmaceutical companies deemed it too risky to transport large quantities of yams from Mexico to the U.S. So, Marker quit his job and went to Mexico alone. He hired Mexican workers, dug up 10 tons of yams, and obtained 3 kilograms of progesterone through degradation. Mass production of progesterone was finally achieved. It shows that besides unconventional creativity, developing a new drug also requires persistent patience from drug hunters. This is the second factor we'll discuss. The anti-rejection drug sirolimus is the result of American biologist Søren Segal's 25 years of persistence. In the 1970s, Segal wanted to develop a new drug to treat fungal infections like athlete's foot. He discovered a microorganism in the soil of Easter Island, Chile, that secretes a compound with antifungal properties. Segal named this compound sirolimus. During animal experiments, he found that sirolimus could wipe out all harmful fungi, but its side effect suppressed the animal's immune systems. Therefore, his company, American Arist Pharmaceuticals, decided to ditch this drug, but Segal didn't want to give up. He believed that although sirolimus suppresses the immune system, for patients who've had organ transplants, it's an ideal drug because it prevents the patient's body from rejecting the transplant. The pharma company's top brass weren't interested in organ transplant drugs and shot down Segal's idea. Well, but Segal was savvy. He knew big pharma execs change often, so whenever a new management team took over, he'd propose testing sirolimus as an anti-rejection drug. After doing this 3 or 4 times, his boss got fed up and ordered him to toss sirolimus's culture into an autoclave to destroy it. Segal pretended to obey, but secretly took a sample home. Later, his boss moved to a different role and a new management team took charge of the company. Segal proposed testing sirolimus as an anti-rejection drug once more, and this time the project got the green light. Follow-up animal and human tests were super successful. By 1999, 25 years after Segal first discovered sirolimus, this antifungal drug was finally approved by the FDA as an immunosuppressive drug. It's now one of the commonly used anti-rejection meds. On the path to finding new drugs, besides drug hunters' smarts and grit, sometimes outside luck can shift the whole research direction. This brings us to the third factor, like vitamin C. Vitamin C deficiency is a scary disease that's been around forever, causing gum bleeding and body-wide hemorrhaging that can lead to death. In the 15th century, as Europeans started long sea voyages, vitamin C deficiency exploded. Healthy, lively sailors would suddenly get sick, with breathing trouble, cracked limbs, skin black as ink, teeth falling out, and mouths giving off a corrosive stench. By the 18th century, no one still knew what caused this disease or how to prevent or cure it. Back then, many sailors on the British Royal Navy fleet got vitamin C deficiency. Ship's Dr. James Lind ran group trials on patients, testing different foods on each group, showing citrus fruits and apple cider vinegar could treat it effectively, but no one could explain why. Scientists were slow to discover vitamin C because they couldn't run proper animal or human tests. Almost all mammals except humans can make their own vitamin C, so we couldn't induce vitamin C deficiency in animals for experiments. But for human tests, no one wanted to suffer through the disease for medical trials, especially since it might not even be curable. But in 1907, a stroke of dumb luck hit two Norwegian scientists studying beriberi.
They fed guinea pigs only grains and flour, hoping they'd get beriberi. But oh wow, the guinea pigs got vitamin C deficiency instead. What a happy accident! Those scientists got super lucky finding an animal that, like humans, couldn't make its own vitamin C.
With animal samples, scientists studied citrus fruits and in 1931 found hexuronic acid, that's vitamin C. Getting a new drug from lab to market needs big clinical trials and mass production, which is impossible without enough cash. So the fourth key factor is funding. Antihypertensive drugs. The birth of Captopril was a capital gamble for the pharma company. In the 1980s, every popular blood pressure drug had nasty side effects. Some caused peeing all the time. Others led to impotence or breathing trouble. Two Squibb scientists, Dave Cushman and Miguel Ondetti, decided to cook up a perfect blood pressure med.
They found rattlesnake venom could drop prey's blood pressure fast, making them pass out, so they pulled out teprotide from the venom. It blocks the ACE enzyme that hikes up BP.
But this drug got wrecked by stomach enzymes if swallowed, which makes sense. If snakes gulp their own venom and gut juice can't break it down, wouldn't they poison themselves?
After hundreds of tries, Cushman and Ondetti nailed an ideal compound like teprotide, but safe from stomach acid, Captopril.
But Squibb execs weren't keen to greenlight the next round of clinical trials. And they believe that even if this blood pressure drug succeeds, it would only add a few $1 million in sales annually, far from covering the high costs of clinical trials and marketing. Cushman and Ondetti didn't want their hard work to go to waste. After getting company approval, they published this research in mainstream journals, attracting unprecedented attention in academia.
Surprised! Successful! Squibb executives eventually approved Captopril's clinical trials. Captopril passed smoothly and gained FDA approval in 1981.
After the drug hit the market, first-year sales exceeded $1 billion, with profits surpassing all other squid drugs combined. As for the industrial mass production of drugs, we have to mention the capital support for penicillin. In 1928, British bacteriologist Alexander Fleming extracted penicillin in his lab. Penicillin's bactericidal effect is remarkable. It can kill many pathogenic bacteria and cure various diseases caused by bacterial infections. In 1939, British scientists Howard Florey and Ernst Chain tested higher purity penicillin and found it worked faster and stronger. They tested it on a patient with a severe bacterial infection. Penicillin worked, but the dose was too small, and the patient ultimately died. Florey. They realized that large doses of penicillin were key to treating bacterial infections, but mass-producing penicillin was a big challenge. At that time, during World War II, Britain was fighting Nazi Germany with uncertain outcomes and lacked the ability to use limited industrial resources for drug production. So Florey, with coordination from the Rockefeller Foundation, went to the U.S. to seek funding. The U.S. Department of Agriculture and multiple large pharmaceutical companies participated in funding. Innovative scientific breakthrough. Scientists at the Agriculture Department found that using corn steep liquor as a culture medium could increase penicillin production 20-fold. They also found a new strain from a moldy cantaloupe, boosting penicillin production 1,000 times more. Afterward, 4 renowned American pharmaceutical companies, including Pfizer, Merck, and Squibb, participated in developing and producing penicillin. At that time, penicillin production was still low. Dozens of American factories and thousands of engineers collaborated, increasing penicillin concentration nearly 100-fold, achieving large-scale mass production. The penicillin project was the second most important project in the U.S. during 281, after the Manhattan Project, receiving huge support from government to industry. Total investment exceeded $5 million. By 1944, penicillin production could meet all Allied forces' demands. Penicillin's dramatic emergence turned the tide for the Allies in World War II. Post-war, penicillin saw wider application, saving tens of millions of lives. Truly, developing a new drug requires drug hunters' creativity, patience, luck, plus external financial backing. But even so, historically successful drug development odds are low. Most attempts fail. So, what challenges does drug development face? And what about its future?
That brings us to part 3. Developing an effective drug requires 2 conditions. First, a suitable compound, the drug itself.
Second, a suitable target, a protein treatable with drugs. If scientists want to impact health, say easing depression or treating food poisoning, they must first find target proteins affecting physiological processes. But the human body is an incredibly complex molecular system with ever-changing relationships. Scientists still can't map how most body molecules work, let alone account for each person's unique genetics and physiology. Plus, despite growing knowledge of cells and organs, we still can't predict how a given compound will react with a human molecule. Worse, most times, there's no given compound. Potential drug compounds in the universe number 3 times 10 to the 6th.
How big is that number?
Imagine testing 1,000 compounds per second for breast cancer drugs. By the time the sun burns out, you'd only scratch 3 times 10 to the 6th times squared surface. Typically, only 5% of pharmaceutical research projects get management approval. Of those approved, just 2% yield FDA-approved drugs, meaning overall success is a mere 0.1%. Modern drug researchers tirelessly sift through countless compounds, spending lifetimes seeking substances that can change human destiny. Drug development challenges lead to soaring costs. Each FDA-approved drug averages $1.05. In 14 years, many big pharma projects fail after billions invested with zero returns.
When pricing new drugs, companies must recoup countless failures into few successes, causing astronomical prices.
Even then, new drugs get under 10 years of patent protection.
Pharma's exclusive high-profit window is short. Once patents expire, cheap generics flood the market like mushrooms after rain.
Additionally, the author notes high costs discourage developing curative drugs.
Because if a drug cures completely, patients won't repurchase, leaving minimal profit margins.
Antibiotics have slim profits. One course cures patients. Vaccines are worse off.
Vaccines are public health drugs, often involving governments, with smaller profits. The immense challenges make big pharma increasingly hesitant. The author writes that Pfizer executives considered exiting R&D entirely, just buying existing drugs.
That Pfizer, oldest, most talented, wealthiest, and largest pharma company, shows how daunting drug development truly is.
So where's the future of drug development? With 35 years' experience, the author states, creativity remains pharma's truth despite uncertainty. Only when scientists get creative freedom without unnecessary interference can we create continuous, life-changing medicines. And on the drug discovery path, some falter at obstacles while others charge ahead, cutting through thorns. To frontline drug hunters, eternal admiration. That concludes our interpretation of the drug hunters. Notes below. Until next time.